Hey guys, welcome back. So last time we talked about meiosis, cutting the chromosome number in half, and today we're going to take a little closer look at how exactly we do that. And so here's your phases of meiosis. The nice thing about following mitosis with meiosis is they're very, very similar. Here we have interphase, where we replicate our DNA, just like in mitosis. Then we go to meiosis 1, which is prophase, metaphase, anaphase, telophase. We just end them all with 1. And without any interphase here, we roll straight into meiosis 2, which is prophase 2, metaphase 2, anaphase 2, telophase 2. And so we double, we split again, and we end up at half. So the two divisions in meiosis splits our chromosomes from our 2N nucleus into 4 of our haploid nuclei, which we've said before. In meiosis 1, each duplicated homologous chromosome is separated from its partner, and in meiosis 2, we separate our sister chromatids. And so here's the way this looks. Perhaps this is, chromosome 1 from mom, and in the back, chromosome 1 from dad, and they duplicate, and then we have chromosome 1 from mom, chromosome 1 from dad. So in meiosis 1, we're going to separate this. We got mom's chromosomes, dad's chromosomes. In meiosis 2, we separate them again. Mom's chromosome in one cell, the other copy of mom's chromosome in one cell, one copy of dad's in one cell, other copy of dad's in one cell. And what you end up getting is these are now in a different combination because this happened in chromosome 1, and the same thing happened in chromosome 2 and 3 and 4. And so what combination of mom's and dad's copies you're going to get for each chromosome is going to be random. So fertilization functions to restore our chromosome number. The diploid chromosome number is restored at fertilization because we have 2 haploid gametes, 1 egg, which is 1N, 1 sperm, which is 1N, fused to form a zygote. And a zygote is a single-celled, brand new, little tiny individual. And that's kind of cool to think about, the first cell of a new individual, because everything you see around you, trees and animals and ourselves, we all started as one cell, one teeny tiny zygote with enough information to differentiate and develop into the adult form. Without meiosis before fertilization, we would have this problem of doubling a chromosome number with every single generation, and that would probably be pretty bad. Humans and animals certainly couldn't tolerate this. Plants could tolerate it for a little bit, give or take, but eventually they wouldn't be able to tolerate it either. And so meiosis maintains our chromosome number, and hopefully by mixing around those genes, or rather those chromosomes, it maintains our organism health because it gives us a lot of genetic diversity. Alright, here's a fun title. The visual tour of meiosis. I don't know if it's gonna shape up to be as much fun as it sounds, but there you go. We'll get into it at any rate. Meiosis cuts our chromosome numbers in half. We've already said that. Chromosomes of a diploid nucleus become distributed to 4 haploid nuclei. We've already covered that. It's in 2 stages, 1 and 2. We've already covered that. Let's jump right into it. Prophase 1. Just like in mitosis, our chromosomes condense and become visible. They pair up, and then unlike mitosis, they're actually able to swap segments, which is kind of cool. We call this process crossing over, and it's going to make the genetic diversity that we could accomplish before about, I don't know, 1,000,000 times more complicated and more diverse because of crossing over. So they condense, they pair up, they cross over. Spindle microtubules attach to condensed chromosomes, and our nuclear envelope breaks up, just like in mitosis. Metaphase 1, our homologous chromosomes align in the middle, just like in mitosis. Our spindle attaches to the chromosomes, getting them all lined up, just like in mitosis. So it looks pretty familiar, right? Prophase 1, we had a little bit of crossing over right there. Metaphase 1, they're in the middle, so we had visible chromosomes. Now they're lined up chromosomes. Anaphase 1, you know what happens in anaphase already. Our chromosomes are going to separate, head towards opposite poles. Telophase 1, the sets of chromosomes are clustered at both ends of the cells, and we go ahead and form a new nuclear envelope around each set, and we have 2 haploid nuclei. Alright, so here you have it, anaphase, we pull them apart. Telophase, we've got nuclear envelope, copy of dad's, copy of mom's, and that's where we're sitting. And so here's what they look like. Anaphase being pulled apart, telophase, they're pretty condensed. Now here's where this becomes very, very, very different from what we've learned before. We don't go to interphase, we don't duplicate it, or we would just be doing mitosis twice. We jump straight into prophase 2, where our chromosomes condense, again, our microtubules attach to sister chromatids as the nuclear envelope breaks up, again. Metaphase 2, they align in the middle, again. Okay, so here we have prophase 2, you can see condensed chromosomes. Metaphase 2, they're lined up in the middle, this is occurring again. Anaphase 2, they separate again. Telophase, they cluster at the end, and we get a nuclear envelope again. And so look what we've done at the end. 1 cell, 2 cells, 3 cells, 4 cells. Each one has 1 copy of mom's with a little bit of crossing over from dad's, and 1 copy of dad's. Okay? And bear in mind, as I'm using these terms, mom and dad, I guess you'd have to think of it this way. If you're a human making 4 sperm cells, this would be 1 copy of your dad's and 1 copy of your mom's. And if you're a human making egg cells, this would be, 1 copy of your mom's and 1 copy of your dad's. And so for the new kiddo who's going to be formed, for your kiddo, what they're getting is a unique shuffling of most of grandma's chromosome with a little bit of granddad's chromosome mixed in there, and an entire set of granddad's chromosomes. Or all of granddad's chromosome 1 and all of grandma's chromosome 2. And so you can see that they become very, very, very diverse, because not one of these is alike. And this little cartoon's only with 2 chromosomes. You can imagine doing this with 23 chromosomes and a sex chromosome, or 22 chromosomes and a sex chromosome, that the combinations would just be ridiculous. So we're going to talk a little bit about variation. Variation traits is a good thing. You probably don't look exactly like your mom or exactly like your dad. And if you have brothers and sisters, you guys might not even look like an even blend between mom and dad, because there's a lot of variation from meiosis. So crossing over and the random sorting of chromosomes is going to give us new combinations. Random sorting is a very important concept. And basically what it means is there's nothing that says that dad's chromosome 1 has to go one way and mom's chromosome 1 copy has to go another way. These get completely mixed up, and it's random. And so that's going to make a lot of diversity for our offspring. And along with fertilization, these contribute to variation among the offspring of our sexually reproducing species. And so where we really make this more diverse is with crossing over. That's in prophase 1, and a chromosome and its homologous chromosome next to it can sometimes exchange these corresponding segments, which is what I showed you in that last picture where I said you had a little bit of granddad's chromosome on grandma's chromosome. Crossing over occurs in prophase 1, and what you get is when these 2 chromosomes line up and kind of overlap each other in a place where they're carrying the same gene, they can actually pass that gene to a different chromosome in exchange for that chromosome's copy of the gene. And so you get a unique blend of alleles that your parents carried from their parents. and those have been shuffled and randomly distributed with each generation. And so it's probably pretty likely that your aunts and uncles don't look alike either. So here's what this looks like. We've got one chromosome. They went ahead and marked alleles to make it easier for us. This is a gene, a chromosome from dad, chromosome from mom. We've got a big A allele and a big B allele for the gene that they're carrying. Now mom's got a little A allele and a little B allele for the gene that she's carrying. Now, when meiosis occurs, it just so happens that these two don't cross over, so they maintain the exact same alleles they did before. And it just so happens that this one here does cross over. Okay, so I guess it has the ability to cross over. And so what you get is these two overlapping. And when they do that, we're going to break a little end off of mom's and attach it to dad's. Or break a little end off dad's and attach it to mom's. So now mom's chromosome carries a little A allele and a big B allele, and a little A allele and a little B allele. And so now mom's chromosome has the ability to pass on to you and express a trait that would not have formerly been expressed by this chromosome. And the same goes for this one. And so when you add this in for 22 chromosomes, and you consider that there's more than one location that crossing over can occur at, the cartoon makes it 1 to make it simple, but there's nothing that says it has to be 1. Then the diversity becomes pretty mind-boggling. Here's a picture of what it really looks like. This is kind of cool. A centromere crossing over, more crossing over. So when they're pulled apart, they get different strands of a chromosome. So that's kind of cool. Alright, so chromosome segregation, when you want to split them up. Homologous chromosomes can be attached to either spindle pole in prophase 1, and so each homolog can be packaged into either one of the two new nuclei, which means there's no rule that says this chromosome copy here has to go this way, and this chromosome copy here has to go this way. That's completely random, and we're going to randomize it for all 22 chromosomes and one sex chromosome. So what you end up getting is you say to yourself, Mr. Stecker, I want to have enough kiddos that by random chance I have two kiddos who are not monozygotic twins who have the same sets of alleles from random segregation in meiosis. How many kiddos do I need to have? And the answer is somewhere in the order of 8,388,608 kiddos if you're a human with 23 chromosomes. That's how many children you'd have to have before two of them had the same combination of genetic material, statistically, anyways. So there's that. That's a lot of variety. Okay, probably don't look like your brothers and sisters exactly, because you got a lot of variety. Okay, so here's some 4 possible alignments. They went ahead and put metaphase 1. All the blues happen to be on this side. All the reds happen to be on this side. Here we got 2 blues and a red, 2 reds and a blue. Here we got 2 reds and a blue. And here we got 2 different reds in the middle blue. So they're just randomly organized at metaphase. When we split them up, we get random combinations of maternal and paternal chromosomes. And then when we split those up, we get random combinations down here. And so each of those eggs, or each of those sperm, or each of those pollen grains are going to be unique and distinct. So, mitosis and meiosis. Your book leaves us with this idea towards the end that they're so similar. You know, certainly one must have evolved from the other. I'll let you kind of chew that idea over, and we'll talk about it maybe a little bit in class, but the argument is, you know, though they have different results, mitosis and meiosis are fundamentally very similar. Meiosis may have evolved by the remodeling of existing mechanisms of mitosis, because meiosis is a lot like doing mitosis twice. So maybe it started that way. Mitotic divisions produce clones. Meiotic divisions produce offspring that are different. And so despite the different end results, their stages are very, very, very similar. And so here you have mitosis, prophase, metaphase, anaphase, telophase. We made clones. Here you have meiosis too. Prophase, metaphase, anaphase, telophase. Haploid gametes. So they are pretty similar. Here's a cool example of your BRCA genes coming back that we just learned about last chapter. Our molecular products of the BRCA genes monitor and repair breaks in DNA. We learned about these guys in cancer. So for example, during DNA replication before mitosis, the same molecules that monitor and fix the breaks in homologous chromosomes do this during crossing over of prophase 1. And so this guy has a job in mitosis, but he also has a job in meiosis. He fixes DNA during DNA replication, and he will fix our homologous chromosomes once they've done crossing over to make sure everything's pieced together. And so, is that evidence of evolution from meiosis coming from mitosis? I don't think so, but I don't know. We can talk about it in class, I guess. It's cool. Don't get me wrong. I think that's neat that he has that many jobs. But I don't know. It doesn't do anything for me, I guess. So if you have mutations in the BRCA genes, in their molecules, they can affect both the outcomes of mitosis and meiosis. No duh. I'm sorry. Like, you have a mutation in spell check that could affect both a Word document and a note. Yep, because it's still spell checking. That's the same thing, right? Cellular reproduction probably originated by mutations that affected mitosis. Sure. Remodeling existing processes into new ones is a common evolutionary theme, and we'll take a good hard look at that later in the year, so I can go with that. Here's your picture of MLH1 from your book. The blue dots are the centromeres, and the yellow dots are these MLH1 proteins that are maintaining the ends of these chromosomes. So those are kind of a cool picture. Here's the end of the chapter. Why do cells undergoing mitosis require one set of divisions, but cells undergoing meiosis need two? Why does crossing over occur in prophase of meiosis and not in mitosis? And lastly, why is sexual reproduction considered a hallmark of evolutionary advancement? It's a nice, what a way to word that. I love it. Okay, cool. Well, there's chapter, 12 for you, meiosis. So thanks for your time.